today I'm going to present one of my postdoctoral work, which was showing how niche-derived signal induces phenotypic plasticity in intestinal secretory cells. So the intestinal epithelium is a very specialized tissue. It's a monolayer of epithelium that acts as a barrier against the invading pathogen and the gut lumen. And it's also a very rapidly renewing tissue. We know that the stem cells located at the base of the crypt, which are LGR5 positive, can give rise to absorptive enterocytes, or it can give rise to distinct secretory cells that perform various antimicrobial functions and also secrete hormones. Now, through knockout studies and imaging, we know that the intestinal epithelium 801 transcription factor drives the secretory differentiation, and downstream to 801, we have transcription factor neurogenin 3 that drives the enteroendocrine cell specification, and we have PAU2F3 that drives the tough cell specification. However, the two most abundant secretory cell types, goblet and Paneth cell, the transcriptional basis for this cell type is still unclear. And we also wanted to know the chromatin dynamics happening when the stem cells is differentiating towards the secretory fate. So to address this question, the following project ensued, and to specifically enrich for rare secretory cell types, we utilized the 801 Cree rosa tomato mouse model, where upon tamoxifen induction, any cell which is expressing 801 will be permanently labeled in red. So to specifically enrich for early progenitor cells, we performed 2 short doses of tamoxifen and harvested the crypt cells and isolated the tomato positive cells from the duodenum and the ileum of the mouse intestine and performed single cell RNA sequencing. As you can see here, we were able to enrich for more than 20,000 cells from the duodenum and ileum, and it was enriched for secretory cells which were in their differentiation continuum. And now we can ask the question, what are the transcription factors that are enriched in these cell types? So if you look at the right side in the enteroendocrine cells and the tough cells, we see a distinct enrichment of transcription factors. Here I'm just showing top 5 transcription factors, but there were many more. But if you come back to the goblet and Paneth cluster, we don't see any unique enrichment for transcription factor per se. And we also validated this finding through bulk RNA sequencing because we know that the single cell RNA sequencing has very high dropout rate. Since there was lack of distinct transcription factors in these cell types, we asked, how do the goblet and Paneth cells differ in their transcriptome? If you look at the global profile here too, the goblet and Paneth cells are very similar compared to the other secretory cell types. If it's lighter in color, that means it's more similar. And if it's darker, that means it's more distinct. And if one does a pairwise comparison of goblet cells with other secretory cell types, we see that Paneth cells are enriched for very few secretory antimicrobial genes specifically. And this phenomenon is also true in human cells. Not only in RNA level, but also in protein level, if one stains for MUC2, which is a classic goblet cell marker, we see that Paneth cells in the base of the crypt also express them in lower levels. And we also occasionally find goblet cells in the crypt region, which is staining positive for lysozyme, which is a classic Paneth cell marker. And also during the course of our analysis, we can't help but notice that Paneth cells had 2 to 3 times more mean RNA counts, and more than 50 to 60% of the mean RNA counts were coming from the top 10 transcript. And among them, the top 2 transcript, defensin alpha 24 and defensin alpha 30, accounted for more than 50% of the reads. And this also skews the normalization in the downstream analysis of the single cell RNA. And we also validated this finding in previously published single cell RNA data that this data is indeed reproducible. So this overall lack of transcription factor enrichments in goblet and Paneth cells and this skewing of hypertranscription of few specific antimicrobial genes led us to hypothesize maybe Paneth cell is a variant of a goblet cell that hypertranscribes antimicrobial genes. And let me remind you, transcription factor also modulate the chromatin profile, and we were also interested in understanding the chromatin dynamics during the secretory cell differentiation. So to address this, we performed single cell ATAC-seq with the same timeline of that of the single cell RNA. And here too, we were able to efficiently capture all the secretory cell types. And then we asked the question, how do the cell types differ in their global chromatin profile? So here again, we see that like RNA, goblet and Paneth have a very similar open chromatin profile. If it's more red, it's more similar. And if it's more blue, it's more distinct. And we see that the EC and Tuft, which had enrichment for specific transcription factors, has a very distinct chromatin profile. And previous studies from our lab have already shown that the enterocytes and ISC have a very overlapping chromatin profile, and this we validated here as well. What about the enriched enhancers in this specific cell type? Just like transcription factors, it was also very difficult for us to find any enriched enhancers in goblet and Paneth cells. Among the 715 enhancers we saw enriched in goblet cells, these were also accessible in other cell types. In Paneth, we had two varieties of enriched enhancers. Half of them were shared among other cell types, and the rest was unique to Paneth cells. And these were enhancers which were close to the antimicrobial genes. And now if one comes to the enteroendocrine cells or tuft cells, you can easily see around 3,000 to 4,000 enriched enhancer sites. Now, if we zoom into particular locus, MUC2, which is a goblet gene, we see that the Paneth cell also have accessibility in MUC2 locus. And again, if you go to the classic MMP7, it's a Paneth cell marker, we see not only it's accessible in Paneth cell, also goblet cell and its precursor cell types have accessibility. If we come to antimicrobial genes, they show unique accessibility in Paneth cells. And if you go for tuft or EC specific genes, they also have this unique accessibility. Next, we ask the question, what motifs are enriched in these unique enhancers of the Paneth cells? And we saw that these were predominantly TCF-left motifs, highlighting that it might be dependent on the Wnt signaling. And now from the anatomy, we know that the Paneth cells reside in the base of the crypt, which is a Wnt high, BMP low environment. And all the differentiated cells, including the goblet cells, usually migrate upwards and goes to the BMP high, Wnt low environment. Thus, we hypothesized maybe goblet and Paneth cells may be a phenotypic variant of the same cells, which is taking different phenotypes depending to the niche where it finds itself. And indeed, when we look at the transcription factors which are downstream to BMP signaling like ID1 and ID3, it was uniquely enriched in goblet cells. And SOX9, which is a downstream transcription factor for Wnt signaling, it was uniquely enriched in Paneth cells. Then when we read up some literatures, we saw that it's already known in the field that the Wnt signaling induces Paneth cell differentiation. But with our understanding and hypothesis, we hypothesized that the Paneth cell differentiation is not only induced by Wnt signaling, there is also a goblet to Paneth switch, which is happening due to the Wnt signaling. And we performed series of experiments. Here I'm just showing one representative image where we acutely induce high Wnt scenario in 81 positive cells by knocking out APC. Here you can see the classic goblet cells with goblet morphology staining positive for lysozyme, which is a classic Paneth marker. But whenever we did this experiment, we never found this phenotypic switch in the tip of the villus. And we know that in the tip of the villus, the mesenchymal cells here secrete high levels of BMP. And this led us to hypothesize maybe BMP signaling might be a potent inhibitor of Paneth phenotype. So to address this question in vivo, we utilized GRIM1 DTR mice. So just below the intestinal crypt where we have the Paneth cells, we have mesenchymal cells that secrete gremlin-1, which is a BMP inhibitor. They also secrete Wnt coactivator R-spondin. So when we add the diphtheria toxin, these gremlin-1 positive cells will be killed, and it will increase the BMP tone in the crimped region. And we see that upon ablation of these gremlin-1 positive cells, we see a gradual decrease of Paneth cell marker, which is highlighted in brown, and we see increase in Alcian blue staining of a goblet cell marker. Next, to conclusively show that this is indeed a phenotypic switch from Paneth to goblet in vivo, we again went back to our 801 Cree rosa tomato mice. We injected tamoxifen and waited for 2 weeks. So in these 2 weeks, what happens? All the differentiated cells migrate upwards and are lost from the tip of the villus, but the Paneth cells go down, migrate down, and they are staying there for 60 to 100 days. And we can see here as well. So next, what we did, we decreased the tone of the BMP signaling by injecting mice with antibody against R-spondin 2 and 3. And within 40 hours, we see that these Paneth cells are changing their phenotype. They take the goblet morphology and are positive for the goblet marker MUC2. Now, next, to mechanistically show how this goblet Paneth phenotype is being switched and to mimic the in vivo environment of Wnt high BMP low and BMP high Wnt low, we switched to the organoid model. And here what we did is we treated the organoids with DAPT, which is a notch inhibitor, and it induces secretory cell differentiation and also stops proliferation of the cell. So whenever we do any treatment, it's the cells which we have to begin with, and they are taking that phenotype. And again, here we see that Wnt high BMP low scenario induces Paneth phenotype as known, but what's striking here is that the BMP high Wnt low scenario decreases the Paneth genes by orders 2 to 3 orders of magnitude. And also you can see it in the protein level. And whatever finding we had here had a chromatin underpinning in BMP high scenario. We see that the goblet specific loci opening up, and PANET-specific loci going down. Not only that, we can also in organoid system convert the cells to the PANET phenotype with Wnt high, BMP low, and then again treat those cells with BMP high, Wnt low scenario, and they take up the goblet profile. And here are some representative RNA genes and immunostainings. Finally, we ask the question whether this finding can be also extended to humans. So to address that, we developed a DOX-inducible 801 construct, and we transfected the cells, primary human ileal cells with that. So upon doxycycline induction, the cells will start making 801, and this will be MHRE positive. And we see that within 2 days, an efficient induction of secretory genes as evident from here. And again, then we treat with the BMP high scenario or BMP low Wnt high scenario, we see it takes up either the goblet phenotype or the PANET phenotype. So to overall tell you, the story I presented here is that the goblet and PANET cells show limited chromatin and transcriptional diversity. And these are not distinct cell type per se, like the enteroendocrine cells and tuft cells. These are a variant of the single 801 positive secretory cells, which in BMP high Wnt low scenario takes the goblet phenotype, and Wnt high BMP low scenario takes the PANET phenotype. If you're interested to know about this story more in details, you can read in this paper. And finally, I would like to thank my mentor, Ramesh, and my wonderful lab members and collaborators and the funding agencies. Thank you, and I look forward to any questions you may have.
Great, thank you very much. Very interesting talk. A virtual round of applause for you as well. We have a couple of questions in the Q&A box already. So the first question is from Anonymous. What do you think is driving the PANIS-cell migration down right after cell fate commitment? I.e., what niche factor, cell-to-cell interaction or niche-to-cell interaction are involved?
So it might be related again to the Wnt signaling. There was a classic paper by Hans Klever's group where they show the F-RIN F signaling pathways, and that plays the boundary between what stays below the crypt and what goes up the crypt. So predominantly Wnt signaling not only driving the PANET phenotype, but it also might be keeping them in below the crypt region.
Okay, thank you. And a second question from the same A10D. Is there any specific transcription factors or epigenetic oscillation stage during the goblet to PANIS cell fate commitment process, just like EEC differentiation?
Yeah, that's a great question. So we have not looked into that, but what we see that there might be group of transcription factors which might be working, since we didn't see any uniquely enriched transcription factor, but there might be some on lower level. They might be oscillating or going through up and down, and that might be the case, but we haven't explored that possibility in this paper.
So I have a question. It's about whether your transcriptome data also included non-coding RNAs and whether there might be an element of non-code, since transcription factor profiles and chromatin profiles are quite similar, whether there's another level of regulation happening there.
Yeah, no, that's a great question. We have not looked at the non-coding RNA in this particular study.
Sure. Any other questions? I'll give people a few seconds to type their questions in if they have any. Another question that I had, and you did show at the beginning, but I didn't look in the same context, is whether you see any evidence of this transdifferentiation between cell types in your pseudotime analysis. Do you see goblet cells and PANIS cells and something in between those two cell
So,
types?
so first of all, I would maybe not say that this is a transdifferentiation and this is
Okay,
maybe a phenotypic switch. This is the
sure.
same cell. Transdifferentiation usually happens when a particular cell type changes to completely different cell type.
Hmm.
But in historically, if you do single cell RNA sequencing of intestinal epithelium, goblet and PANET cells always cluster together.
Yeah.
So that's also one indication that they might have a shared identity. And again, it might be that there is a common precursor cell, which I'm saying that if you put them in high BMP, it takes goblet, or
Hmm.
put them in high Wnt, takes the PANET phenotype. So it might be, there might be an intermediate state in between, but we haven't explored that a possibility there. So it might not be direct goblet to PANET, but through an intermediate early precursor state.
Sure. Okay, thank you very much.